Hey there, science fans! Today, we're diving into the fascinating world of gamma radiation and its effects on our blood. Specifically, we're looking at hemoglobin, the protein in our red blood cells that carries oxygen throughout our bodies. It's a critical component of our respiratory system, and understanding how it's affected by radiation is crucial for fields like medicine and space exploration. Now, gamma rays are a type of high-energy electromagnetic radiation. They're emitted by radioactive materials and pack a punch. When these rays interact with living organisms, they can cause a lot of damage at the cellular level. That's why it's so important to study their effects and find ways to protect ourselves. In this study, scientists wanted to see how different doses of gamma radiation affected the structure and function of hemoglobin. To do this, they used a group of adult male albino rats as their subjects. Rats are often used in scientific research because they have a similar physiology to humans. This research helps us understand the risks associated with radiation exposure. It also paves the way for developing new treatments and protective measures. So buckle up as we explore the intricacies of this important scientific investigation. Let's go! To make sure the experiment was fair and accurate, the scientists divided the rats into several groups. Each group received a different dose of gamma radiation, carefully measured in units called grays, GY. One group, the control group, received no radiation at all. This group served as a baseline for comparison. The rats were exposed to the gamma radiation in a controlled environment. This ensured that all the rats in a particular group received the exact same dose. Just like a photographer uses a light meter to measure the brightness of light, scientists use special instruments to measure radiation levels. The scientists carefully monitored the rats throughout the experiment, observing their overall health and behavior. This helped them understand any immediate effects of the radiation exposure. But the real investigation began after the irradiation was complete. By using different groups of rats and varying the radiation dose, the scientists could study the relationship between exposure and its effects on hemoglobin. It's like adjusting the volume on a speaker to see how it changes the sound. This approach allowed for a more comprehensive understanding of gamma radiation's impact. After irradiation, scientists collected blood samples from the rats. Using a fine needle and small tube just like at the doctor's office, the samples held the key to understanding gamma radiation's effect on hemoglobin. They separated hemoglobin from other blood components like separating cake ingredients. Techniques included washing, lysing, and centrifuging. Lysing broke open red blood cells to release hemoglobin. Centrifuging separated heavier components from the hemoglobin solution. Pure hemoglobin samples were analyzed using a UV visible spectrophotometer. Stay tuned to see how this instrument helps unlock the secrets of gamma radiation's impact on hemoglobin. Section 4. Shining a light on change. The role of the UV visible spectrophotometer. Now, to understand how gamma radiation affected the hemoglobin, the scientists needed a way to see it. That's where the UV visible spectrophotometer comes in. This instrument shines a beam of light through the hemoglobin solution and measures how much light passes through it. It's like shining a flashlight through colored cellophane. Different colors let different amounts of light through. Hemoglobin, like many molecules, absorbs light at specific wavelengths. This creates a unique fingerprint called an absorption spectrum. By comparing the absorption spectra of hemoglobin from the irradiated rats to the control group, the scientists could identify any changes caused by the gamma radiation. Imagine looking at two fingerprints, one slightly smudged. The spectrophotometer helps us see those smudges and the hemoglobin's absorption spectrum caused by radiation. The greater the changes in the absorption spectrum, the more significant the impact of the radiation on the hemoglobin structure. This analysis allowed the scientists to correlate the different doses of gamma radiation with the degree of change in the hemoglobin. It's like connecting the dots to see the bigger picture. The spectrophotometer served as their high-tech magnifying glass, revealing the hidden effects of radiation at the molecular level. Section 5. Beyond the numbers, unveiling the implications. Scientists used statistical methods to determine the significance of their findings. They wanted to ensure changes were due to gamma radiation, not random chance. One tool used was the student's t-test. It compared average changes in hemoglobin between irradiated and control groups, like comparing average height in two classrooms. The t-test determines if differences are significant or due to natural variation. Results showed a link between gamma radiation and hemoglobin changes. higher radiation doses led to more significant changes, confirming gamma radiation can alter this vital protein. This research has far-reaching implications. It helps protect those frequently exposed to radiation, like astronauts and medical professionals, paving the way for new treatments for radiation sickness and cancer. Unraveling gamma radiation's impact leads to a safer, healthier future. Welcome to the For Science Salah Lotfy channel. Today, we delve into a fascinating research topic that bridges biology and physics, exploring how gamma radiation impacts hemoglobin, the molecule that powers oxygen delivery in our blood. This groundbreaking study, titled Radiation-Induced Changes in the Optical Properties of Hemoglobin Molecule, comes from the Molecular Biophysics Lab at the National Center for Radiation Research and Technology in Cairo, Egypt. The research delves into how radiation exposure alters the optical characteristics of hemoglobin, a crucial protein in our blood responsible for oxygen transport. This study is pivotal in understanding the broader implications of radiation on human health and could pave the way for new medical advancements. The research was led by Professor Dr. Nabila Selim and Associate Professor Dr. Seham El-Marakby, both renowned for their contributions to molecular biophysics. Their combined expertise and dedication have been instrumental in driving this project forward, showcasing their expertise in understanding the intricate effects of radiation on biological systems. The study utilized advanced spectroscopic techniques to meticulously analyze the changes in hemoglobin's optical properties, providing valuable insights that could influence future research and healthcare practices. Published in the renowned journal Spectrochimica Acta Part A, Molecular and Biomolecular Spectroscopy, this groundbreaking research has made significant contributions to the field. This work underwent a rigorous review process, ensuring its quality and impact. The peer review process involved multiple experts in the field who meticulously evaluated the manuscript. The manuscript was initially received on December 6, 2009, marking the beginning of its journey through the publication process. After thorough evaluation, it was revised on February 11, 2010, incorporating valuable feedback from the reviewers. Finally, the manuscript was officially accepted on February 26, 2010, signifying its readiness for publication and dissemination to the scientific community. The research utilized advanced spectroscopic techniques, including both absorption and emission spectroscopy, to gain a comprehensive understanding of the molecular interactions. These techniques were employed to analyze changes in hemoglobin's optical properties when exposed to gamma radiation, providing detailed insights into the molecular dynamics at play. The findings revealed significant alterations in molecular energy levels and structural properties, indicating a profound impact on the stability and function of hemoglobin molecules. This research sheds light on how radiation can impact the function of this vital protein, potentially leading to new insights into radiation therapy and its effects on human health. This study not only enhances our understanding of radiation's effects on biological molecules, but also contributes to improving safety protocols and medical treatments involving radiation exposure. Thank you for joining us to explore this fascinating topic. Stay tuned for more scientific discoveries here on the For Science Salah Lotfy channel. Remember to like, share, and subscribe to support our journey into the world of science. We begin our journey by diving into the microscopic world within us, a universe teeming with complex machinery that keeps us alive. Each cell is a bustling metropolis, with countless interactions happening every second. Here we encounter hemoglobin, a protein residing within our red blood cells. These cells are the lifeline of our body, coursing through our veins and arteries. Picture it as a tiny molecular cage, its iron core clutching onto oxygen molecules like precious cargo. This cargo is vital for our survival. Hemoglobin's mission is critical to transport oxygen from the lungs, where we breathe it in, to every corner of our body. This journey is essential for our cells to function properly. This delivery of oxygen fuels the very essence of our being, powering the cellular engines that drive our every thought, action, and breath. Without this process, life as we know it would cease to exist. Without hemoglobin, our cells would suffocate, starved of the life-giving oxygen they need to function. This would lead to cellular damage and eventually the failure of vital organs. This intricate dance of oxygen delivery is a testament to the elegance of evolution, a process that has fine-tuned hemoglobin over millions of years. Evolution has crafted this molecule to perfection. Its structure, a marvel of biological engineering, is exquisitely adapted for its life-sustaining role. Every aspect of its design serves a purpose. Yet, like all things in the universe, hemoglobin is not immune to disruption. Various factors can affect its efficiency and functionality. Forces from the vast expanse of space, forces like gamma radiation, can penetrate our protective atmosphere and interact with the delicate machinery within us. These interactions can have profound effects. To understand the potential consequences, we must first understand the nature of this invisible radiation. Only then can we appreciate the delicate balance that sustains life. Gamma rays are like cosmic messengers, carrying energy across vast distances from exploding stars and other celestial events. These rays travel through the universe, often originating from some of the most violent and energetic processes known to science. These high-energy photons, part of the electromagnetic spectrum, are invisible to our eyes, yet they possess incredible penetrating power. They can travel through space and matter, revealing secrets about the universe that are otherwise hidden from view. Unlike visible light, which illuminates our world, gamma rays have the ability to pass through solid objects, including our bodies. This unique property allows them to be used in medical imaging and treatments, but it also means they can be quite dangerous. As these energetic waves traverse our cells, they can leave a trail of molecular disruption in their wake. This disruption can lead to significant biological damage, affecting the very building blocks of life. Imagine a bowling ball crashing through a set of carefully arranged pins. That's the kind of havoc gamma radiation can wreak on our delicate biological molecules. The impact is both powerful and chaotic. While we have evolved some natural defenses against low levels of radiation, intense bursts can overwhelm our cellular machinery. Our cells can only handle so much before the damage becomes irreparable. This is where the story of gamma radiation and hemoglobin intersects. A story that highlights the vulnerability of life in the face of cosmic forces. It serves as a reminder of the delicate balance that sustains us and the powerful forces that can disrupt it. Gamma radiation's interaction with matter can create a cascade of damaging events within our cells. When these high-energy photons collide with water molecules, which are abundant in our bodies, they can knock off electrons, transforming stable molecules into highly reactive entities known as free radicals. These rogue molecules with their unpaired electrons are on a quest for stability, desperately seeking to pair up with electrons from other molecules. In their frenzy, they attack nearby proteins, lipids, and even DNA, leaving a trail of molecular damage in their wake. Imagine a group of vandals set loose in a well-organized library. They begin to rip pages from books, tear down shelves, and create chaos wherever they go. Free radicals like these vandals disrupt the order and function of our cells, potentially leading to a range of health problems. To understand how gamma radiation affects hemoglobin at a molecular level, scientists turn to sophisticated tools like UV visible spectroscopy. This technique allows us to study how molecules interact with light, revealing their unique fingerprints. Imagine shining a beam of white light through a prism. The prism separates the light into its constituent colors, revealing a spectrum of hues. Similarly, when we shine UV visible light through a sample of hemoglobin, the molecule absorbs specific wavelengths, creating a distinct pattern of peaks and valleys. This absorption spectrum serves as a molecular fingerprint, providing information about the molecule's energy levels and electronic structure. By comparing the spectrum of a normal hemoglobin sample to one exposed to gamma radiation, scientists can pinpoint the precise locations of damage and gain insights into how the molecule's function is affected. In a recent study conducted by the Molecular Biophysics Lab in Cairo, researchers used UV visible spectroscopy to investigate the effects of gamma radiation on human hemoglobin. Their findings painted a stark picture of the molecular damage inflicted by this invisible force. The study revealed a decrease in light absorption by the irradiated hemoglobin, suggesting alterations in the molecule's structure. This decrease, evident in the shifted peaks and altered intensities of the absorption spectrum, pointed to disruptions in the delicate arrangement of atoms within the hemoglobin molecule. These structural changes, like cracks in a foundation, can compromise the integrity and function of the protein. The researchers also observed shifts in energy levels within the irradiated hemoglobin molecules, further evidence of the profound impact of gamma radiation on this vital protein. Section 6. Gamma's implications for human health. The damage inflicted upon hemoglobin by gamma radiation has profound implications for human health. Since hemoglobin is responsible for oxygen transport, any impairment to its structure or function can have cascading effects throughout the body. Tissues and organs starved of oxygen begin to malfunction, leading to a range of symptoms from fatigue and weakness to more serious complications like organ damage. In extreme cases, high doses of gamma radiation can lead to radiation sickness, a life-threatening condition characterized by nausea, vomiting, hair loss, and a weakened immune system. Understanding the precise mechanisms by which gamma radiation affects hemoglobin is crucial for developing effective medical countermeasures. By unraveling the molecular intricacies of this interaction, scientists hope to develop treatments that can mitigate the harmful effects of radiation exposure. Section 7. The body fights back. Natural defenses against radiation. While the picture painted so far may seem bleak, it's important to remember that our bodies are not passive victims in this cosmic dance. Over millennia, life on Earth has evolved a range of sophisticated defense mechanisms to repair and counteract the damage inflicted by radiation. Our cells possess an arsenal of antioxidant enzymes like specialized repair crews that work tirelessly to neutralize free radicals and repair damaged molecules. These enzymes act as scavengers, seeking out and disarming the rogue molecules before they can wreak havoc on critical cellular components. Additionally, our bodies have evolved mechanisms to recognize and destroy cells that are too damaged to repair, preventing them from replicating and potentially causing further harm. This intricate network of defense and repair systems highlights the resilience of life in the face of constant environmental challenges. Section 8. Uncharted territory. The future of gamma radiation research. The journey into the world of gamma radiation and its impact on hemoglobin is far from over. This field of study is continuously evolving, with new discoveries and insights emerging regularly. While we have made significant strides in understanding the molecular underpinnings of this interaction, many questions remain unanswered. The scientific community is committed to exploring these unknowns. Future research will delve deeper into the specific mechanisms by which free radicals target and damage hemoglobin, aiming to uncover the precise pathways involved, seeking to identify the most vulnerable sites within the molecule. This will help in pinpointing the exact areas that are most susceptible to damage. This knowledge will be crucial for developing targeted therapies that can protect or repair hemoglobin in the face of radiation exposure. Such advancements could revolutionize medical treatments. Scientists are also investigating the long-term effects of low-dose radiation exposure, which could have significant implications for public health and safety. A topic of significant relevance to medical professionals, astronauts, and others who may encounter elevated radiation levels. Understanding these effects is crucial for their safety. By unraveling the complex interplay between radiation, hemoglobin, and human health, we can develop better protective measures. We can take steps to mitigate risks and safeguard our well-being in an increasingly technological world. The future of gamma radiation research holds promise for groundbreaking discoveries. Section 9. A Universe Within. Our continued quest for understanding. Our exploration of gamma radiation's impact on hemoglobin has taken us on a journey from the vast expanse of space to the intricate workings of our own cells. It has highlighted the interconnectedness of the cosmos, reminding us that we are not isolated beings, but rather integral parts of a much grander scheme. As we continue to unravel the mysteries of the universe, both within and beyond ourselves, we gain a deeper appreciation for the fragility and resilience of life. Each new discovery, like a piece added to an intricate puzzle, brings us closer to understanding our place in the grand tapestry of existence. The quest for knowledge, like the universe itself, is a journey without end, a testament to the boundless curiosity that defines our species. Hey there, science fans! Today we're diving into the fascinating world of gamma radiation and its effects on a vital protein in our blood, hemoglobin. Gamma radiation, as you might know, is a powerful form of energy. It's like the incredible Hulk of the electromagnetic spectrum. Hemoglobin, on the other hand, is the workhorse of our red blood cells. It's responsible for carrying oxygen throughout our bodies, keeping us alive and kicking. This study aimed to understand how different doses of gamma radiation affect the structure and function of hemoglobin. Why is this important? Well, exposure to high levels of radiation can happen in various scenarios like accidents at nuclear power plants or even during some medical treatments. Understanding how radiation impacts our bodies at a molecular level is crucial. It helps us develop strategies to protect ourselves and develop treatments for radiation exposure. So, let's get started and explore the methods and findings of this important research. To study the effects of gamma radiation on hemoglobin, scientists used a group of albino rats. They divided these furry friends into two groups, a control group and an irradiated group. The control group lived their lives in a normal environment, free from any extra radiation exposure. The irradiated group, however, was exposed to varying doses of gamma radiation, ranging from 1 gray, GY, to 7 GY. A gray, in case you're wondering, is a unit used to measure absorbed radiation dose. Think of it like this: 1 GY is like a single zap of radiation energy. The researchers used different doses to see if the effects on hemoglobin changed, depending on the amount of radiation exposure. This is important because it helps us understand the relationship between radiation dose and its biological impact. Now, to study the hemoglobin, scientists needed to get a sample. They drew blood from both the control and irradiated rats. Don't worry though, it was done humanely and ethically, just like when we donate blood. Once they had the blood, they separated the red blood cells, which are chock full of hemoglobin. Think of it as separating the good stuff from the rest. They then broke open the red blood cells to release the hemoglobin inside. This process is kind of like cracking open a piñata filled with precious hemoglobin molecules. Finally, they purified the hemoglobin to remove any other components that might interfere with their analysis. It's like cleaning up the confetti after the piñata party. We want to focus on the prize, which is the pure hemoglobin. Section 4. Shining a light on change, spectrophotometric analysis. With the purified hemoglobin in hand, the scientists used a cool tool called a UV visible spectrophotometer. Now this instrument might sound complicated, but it's really quite simple. It shines a beam of light through the hemoglobin sample and measures how much light passes through at different wavelengths. Think of it like shining a flashlight through colored cellophane. Different colors of cellophane will absorb different amounts of light, right? Similarly, changes in the structure of hemoglobin due to radiation exposure will affect how much light it absorbs at specific wavelengths. The spectrophotometer helps scientists create a sort of fingerprint of the hemoglobin molecule. By comparing the fingerprints of hemoglobin from the control and irradiated groups, they can see if there are any differences caused by the radiation. Section 5. The proof is in the numbers. Statistical validation. Science is all about evidence, and that's where statistical analysis comes in. The scientists used a statistical test called the student's t-test to analyze their findings. Now don't let the name scare you. The t-test is like a detective that helps scientists determine if the differences they see in their results are truly significant or just due to random chance. It's like figuring out if the suspect's fingerprints actually match those at the crime scene. By using the t-test, the researchers can confidently say whether the changes they observed in the hemoglobin of the irradiated rats were indeed caused by the gamma radiation and not just random fluctuations. This rigorous statistical validation is crucial in scientific research because it ensures that the conclusions drawn from the study are reliable and accurate. Section 6. Conclusion. Well, there you have it, folks. We've journeyed into the world of gamma radiation and its impact on hemoglobin. We've seen how scientists carefully designed an experiment using albino rats, prepared hemoglobin samples from their blood, and used a spectrophotometer to analyze the effects of radiation on this vital protein. Remember, research like this is incredibly important. It helps us understand the potential risks of radiation exposure and develop ways to protect ourselves and even treat radiation-induced damage. So the next time you hear about radiation, remember the hard-working hemoglobin molecule and the scientists working tirelessly to unravel the mysteries of the microscopic world. Stay curious, my friends. Hey there, science fans! Welcome back to another exciting episode where we explore the wonders of the human body and the incredible science that keeps us alive. Today we're diving deep into the fascinating world of hemoglobin, the protein that plays a crucial role in our survival and how it's affected by gamma radiation, a form of high-energy radiation. Hemoglobin is like our body's delivery service for oxygen. It's a complex protein found in red blood cells that binds to oxygen molecules in the lungs. This protein, found in red blood cells, picks up oxygen from our lungs and carries it throughout our body, giving our cells the energy to function. Without it, our cells would be starved of oxygen and unable to perform their essential tasks. Think of hemoglobin as a tiny bus carrying oxygen passengers. Each red blood cell is packed with these buses, ensuring that every part of our body gets the oxygen it needs. Without enough buses running, our cells wouldn't get the oxygen they need. This can lead to serious health issues as our cells rely on oxygen to produce energy and maintain their functions. This vital role makes understanding the impact of gamma radiation on hemoglobin crucial. Gamma radiation can alter the structure of hemoglobin, affecting its ability to carry oxygen and potentially leading to harmful consequences for our health. Gamma radiation is a type of high-energy radiation. It's like super-powered sunlight, invisible to our eyes but capable of penetrating deep into matter. While gamma radiation has important uses in medicine and industry, it can also affect our bodies significantly. One key observation is how gamma radiation affects hemoglobin's color. Exposed hemoglobin can become paler, a phenomenon called hypochromicity. Imagine our oxygen buses becoming more transparent after being exposed to this invisible force. This change in color provides clues about how gamma radiation impacts hemoglobin on a molecular level. Scientists study the interaction of light with hemoglobin to understand its properties and behavior under different conditions. One of the key areas of research is how gamma radiation affects hemoglobin. This type of radiation is known to have significant impacts on biological molecules. Through detailed studies, scientists have discovered that gamma radiation changes the way hemoglobin absorbs light. This alteration can be observed in the absorption spectra. Specifically, gamma radiation affects the absorption coefficients, cross-sections, and dipole moments of hemoglobin molecules. These changes can have profound effects on the molecule's function. Additionally, the dipole lengths of hemoglobin molecules are altered, which can influence how these molecules interact with each other and with other biological structures. Think of it like this. Imagine shining a flashlight through a glass of water. The light passes through clearly, illuminating the other side. Now add a few drops of food coloring. The water changes color and the light passing through it is altered. The light passing through changes because the coloring absorbs some of it, altering the light's path and intensity. Gamma radiation is like adding that food coloring to hemoglobin. It changes the molecular structure, affecting how hemoglobin interacts with and absorbs light. This change in light interaction can provide valuable insights into the effects of radiation on biological tissues and help in developing protective measures. Section 4. Energy shifts, unraveling the impact of radiation. Gamma radiation's impact goes beyond just color change. It can also significantly alter the energy levels within hemoglobin molecules. Think of these energy levels like steps on a ladder, each one crucial. Electrons, the tiny particles within atoms, can jump between these steps by absorbing or releasing energy. Gamma radiation can shift these steps, changing the energy gaps and activation energies within the hemoglobin molecule. This can disrupt how hemoglobin functions, potentially affecting its ability to carry oxygen effectively throughout the body. Section 5. Implications. From diagnosis to protection. Understanding how gamma radiation affects hemoglobin has crucial implications for medicine and occupational safety. This knowledge is not just theoretical, it translates into real-world applications that can save lives and improve health outcomes. In medicine, this knowledge helps us use radiation therapy more effectively to target cancer cells while minimizing damage to healthy tissues. By understanding the precise interactions between radiation and hemoglobin, doctors can fine-tune treatment plans to maximize efficacy and reduce side effects. It also guides us in developing treatments for radiation sickness. These treatments are essential for individuals who have been exposed to high levels of radiation, whether through medical procedures, accidents, or occupational hazards. By focusing on the molecular level, researchers can create more effective medications and therapies. In workplaces where exposure to radiation is a risk, such as nuclear power plants and research labs, this understanding helps develop safety protocols and protective gear to safeguard workers. These measures are crucial for preventing radiation-related illnesses and ensuring a safe working environment. Proper training and equipment can make a significant difference in reducing the risks associated with radiation exposure. By studying these microscopic changes in hemoglobin, we gain valuable insights into the broader impact of radiation on the human body, leading to better diagnosis, treatment, and prevention strategies. This research not only enhances our understanding of radiation's effects, but also paves the way for innovations in medical science and occupational health. The ongoing study of hemoglobin and radiation continues to be a vital area of research with far-reaching implications. Hemoglobin is a protein. It resides within our red blood cells. Its job, to carry oxygen. This oxygen fuels our entire body. Without hemoglobin, our cells would starve. We would cease to exist. Hemoglobin's structure is fascinating. It has 4 subunits. Each subunit cradles an iron atom. This iron is what binds to oxygen. Imagine a tiny hand grasping a vital air molecule. This process is essential for life. Oxygen is carried from our lungs. It travels to every corner of our body. Hemoglobin ensures this delivery happens smoothly. Without it, we wouldn't survive. Gamma radiation is energy, pure invisible energy. It travels in waves like ripples in a pond, but these ripples are powerful. They can pass through almost anything. Where does this energy come from? It originates from the atom's nucleus. Nuclear reactions like those in stars produce it. So do certain medical procedures. Gamma rays are ionizing. This means they can knock electrons off atoms. This creates charged particles called ions. These ions can damage our cells. They disrupt delicate biological processes. Our cells have ways to repair some damage, but high doses of gamma radiation are dangerous. They overwhelm our defenses. This leads to radiation sickness and other health problems. Scientists recently studied gamma radiation's effect on hemoglobin. They used advanced techniques to observe the changes. What they found was concerning. One key finding was hypochromicity. This means the hemoglobin became less effective at absorbing light. This suggests structural damage to the molecule. The study also revealed changes in the porphyrin ring. This ring is crucial. It holds the iron atom that binds to oxygen. Damage here could impair oxygen transport. Furthermore, critical bonds within the hemoglobin molecule were altered. These bonds hold the protein together. Their disruption could lead to the molecule unraveling. Section 4. Molecular shifts, a closer look. The study delved into the molecular level. It examined how gamma radiation affected dipole moments. These moments describe the distribution of electrical charge within a molecule. Gamma radiation caused changes in these dipole moments. This suggests a shift in electron distribution within the hemoglobin molecule. Dipole lengths, a measure of charge separation, were also affected. These changes further indicate structural modifications within the molecule. Finally, the study examined energy gaps. These gaps represent the energy required to excite electrons within a molecule. Changes in energy gaps suggest alterations in the molecule's electronic structure. Section 5. Implications for health and safety. These findings have significant implications. They highlight the vulnerability of hemoglobin to gamma radiation. Damage to this vital protein could have serious consequences. Impaired oxygen transport is a major concern. Hemoglobin's ability to carry oxygen could be compromised. This could lead to tissue hypoxia, a lack of oxygen reaching the body's tissues. Furthermore, the study underscores the importance of radiation safety. Exposure to high doses of gamma radiation should be minimized. Proper shielding and safety protocols are crucial. These findings also have implications for medical treatments. Radiation therapy, while life-saving, uses ionizing radiation. Understanding its effects on hemoglobin is crucial for minimizing side effects. Section 6. Conclusion. The unfolding story of hemoglobin. This study provides valuable insights into gamma radiation's impact on hemoglobin. It reveals molecular-level changes that could impair oxygen transport. These findings have significant implications for health and safety. More research is needed to fully understand these effects. But this study serves as a crucial step. It highlights the vulnerability of a fundamental biological molecule to an invisible force. It reminds us of the power of scientific inquiry to unravel the mysteries of our world. It emphasizes the importance of safeguarding ourselves from potential harm. The story of hemoglobin and gamma radiation is still being written. We all know radiation exists. It's a powerful force, both beneficial and dangerous. Today, we're diving deep into how one type of radiation, gamma radiation, affects us at the microscopic level. Specifically, we're looking at hemoglobin, the protein in our red blood cells that carries oxygen throughout our bodies. Understanding how gamma radiation impacts hemoglobin is crucial. It helps us grasp the potential risks of radiation exposure. This knowledge is vital for developing ways to protect ourselves and treat radiation-related illnesses. Studying the effects of invisible forces like gamma radiation can be challenging. These forces are not only invisible, but also incredibly powerful, making them difficult to analyze without specialized equipment. We can't see the changes happening at the molecular level with our naked eyes. These changes are often subtle and require advanced techniques to detect and understand. That's where scientists get ingenious. They develop innovative methods and use cutting-edge technology to uncover these hidden changes. They employ powerful tools like absorption spectroscopy. This technique allows them to see what is otherwise invisible. Imagine shining a light through a prism. The light splits into a spectrum of colors. You see a rainbow of colors, each representing a different wavelength of light. Absorption spectroscopy works similarly, but with much more precision. It shines light through a sample, like hemoglobin, and measures which wavelengths are absorbed. This data is crucial for understanding molecular changes. This absorption pattern acts like a fingerprint, revealing the molecule's unique properties and how they change after being exposed to gamma radiation. This helps scientists understand the profound impact of radiation at a molecular level. Gamma rays are like tiny energy bullets. When they collide with hemoglobin molecules, they can cause significant changes. Imagine a well-organized structure being shaken violently. That's what happens to hemoglobin when exposed to gamma radiation. These changes can disrupt hemoglobin's ability to carry oxygen effectively. Without enough oxygen, our cells and tissues can't function properly, leading to various health problems. The extent of the damage depends on the intensity and duration of the radiation exposure. Unveiling the secret's key discoveries and the path ahead. Through meticulous research using absorption spectroscopy, scientists have made significant strides in understanding how gamma radiation affects hemoglobin. They've discovered that even low doses of gamma radiation can alter hemoglobin's structure, hindering its ability to bind and transport oxygen. These findings are crucial for developing effective countermeasures against radiation damage. But the journey doesn't end there. Advanced techniques like Fourier transform infrared spectroscopy, or FTIR, and Raman spectroscopy are emerging as powerful tools for even deeper analysis. A brighter future. Applications and the quest for knowledge. The knowledge gained from studying gamma radiation's effects on hemoglobin extends far beyond the laboratory. It opens new doors for scientific exploration and practical applications that can transform our world. It has vital applications in various fields, bringing together experts from different disciplines to collaborate and innovate. In medicine, it helps us understand and treat radiation sickness, providing critical insights that improve patient care and outcomes. It also plays a crucial role in protecting patients undergoing radiation therapy, ensuring treatments are both effective and safe. Furthermore, it aids in the development of safer imaging techniques, enhancing diagnostic accuracy and reducing risks. Beyond medicine, this research has implications for space exploration, nuclear energy, and environmental protection. It helps us prepare for the challenges of long-term space missions and the safe use of nuclear technology. As we delve deeper into the universe, understanding the effects of cosmic radiation on astronauts becomes crucial for their safety and mission success. Similarly, managing nuclear waste and mitigating the environmental impact of radiation leaks rely heavily on this knowledge, ensuring a safer and more sustainable future for our planet. In conclusion, A Universe Within revealed. The study of how gamma radiation affects hemoglobin is a journey into the intricate world of molecules and their response to powerful forces. This research delves deep into the very fabric of life, revealing how these tiny components interact and change under different conditions. It's a fascinating exploration that combines biology, chemistry, and physics, showcasing the interconnectedness of all scientific disciplines. It's a testament to human ingenuity and our quest to unravel the universe's secrets one atom at a time. Each discovery, no matter how small, brings us closer to understanding the complex mechanisms that govern our existence. The dedication and perseverance of scientists worldwide drive this relentless pursuit of knowledge. The knowledge we gain from this research not only protects us from harm, but also paves the way for advancements in healthcare, space exploration, and environmental protection. By understanding the effects of gamma radiation, we can develop better medical treatments, enhance the safety of astronauts, and protect our environment from potential hazards. As we continue to explore the vast universe within and around us, one thing remains certain: the pursuit of knowledge and understanding will always be our greatest adventure. This journey is not just about scientific discovery, it's about pushing the boundaries of what we know and inspiring future generations to look up at the stars and wonder. Properties of hemoglobin, a protein essential for oxygen transport in the body. This research, titled Radiation-Induced Changes in the Optical Properties of Hemoglobin Molecule, was conducted at the Molecular Biophysics Lab, Radiation Physics Department, part of the National Center for Radiation Research and Technology in Cairo, Egypt. The primary aim of this study was to explore the intricate ways in which radiation affects the hemoglobin molecule, a crucial component of our blood. By understanding these changes, scientists hope to uncover new insights into the broader implications of radiation exposure on human health. The study delves into how radiation disrupts hemoglobin at the molecular level, using advanced spectroscopy to reveal structural and functional changes. This involves detailed analysis through techniques such as UV visible spectroscopy, which allows researchers to observe the minute alterations in the hemoglobin's optical properties. These findings are crucial for developing better protective measures against radiation. These insights contribute to improving our understanding of radiation's biological effects and its implications for medical and safety applications. By comprehensively studying these molecular changes, the research paves the way for advancements in medical treatments and safety protocols, potentially leading to more effective therapies and protective strategies against radiation exposure. I'd like to extend a heartfelt thank you to the brilliant minds behind this research. Your tireless efforts and unwavering dedication have paved the way for groundbreaking discoveries that will benefit humanity for generations to come. Professor Dr. Nabila Selim and Associate Professor Dr. Seham El-Marakby. These remarkable women have shown exceptional leadership and innovation in their fields. Their dedication and expertise at the Molecular Biophysics Lab of the Egyptian Atomic Energy Authority have provided us with invaluable knowledge. Their work has not only advanced our understanding of molecular biophysics but also opened new avenues for future research. Your efforts in exploring the intricate effects of radiation are truly inspiring and impactful. The meticulous research and collaborative spirit you have demonstrated are a testament to the power of scientific inquiry and the pursuit of knowledge. Now, it's your turn to join the conversation. We've shared some groundbreaking research and fascinating insights, but the discussion doesn't end here. What do you think about the findings of this study? Do you see potential applications or areas for further exploration? How do you believe these results can influence medical treatments or radiation safety? Your insights could spark the next big breakthrough. Share your thoughts in the comments below. Engage with fellow viewers and contribute to this vibrant community of thinkers and innovators. We'd love to hear your perspectives on this incredible research in today's episode. Your voice matters, and together, we can push the boundaries of knowledge. Thank you for watching this episode of the For Science Salah Lotfy channel. Don't forget to like, share, and subscribe for more groundbreaking scientific discoveries. Until next time, stay curious and keep exploring the wonders of science.